Hello everyone. So today I'll be going to explain about cell mediated immunity. So this cell mediated immunity is T lymphocyte mediated and there should be exposure of antigen or else it does not work. There should be exposure to antigen and it provides protection against various viruses, fungi and many other foreign objects which enters the host system. Coming to the types of T lymphocytes before going to the steps of CMI. So there are two types of T lymphocytes, that is CD plus 4 T lymphocyte and CD plus 8 T lymphocyte. And apart from these cells, there is also a major cell called as major histocompatibility complex. And this plays a very major role in presenting the antigen to the T lymphocyte. And this is again of two types, that is major histocompatibility complex 1 and major histocompatibility complex 2. And this MHC1 helps in presenting the antigen to the CD plus 8 T lymphocyte, which is present in all the cells except RBCs. And major histocompatibility complex 2 presents the antigen to the CD plus 4 T lymphocyte, which plays a major role in the immune system and helps in the antigen presenting. For example, dendritic cell. And again, before going to the steps of CMI, it's very important to know about the function of major histocompatibility complex. When an antigen enters into the cell, it gets fragmented. Sorry, I have mentioned it as agenic, it's antigenic actually. So, inside the cell, this major histocompatibility complex will bind with these antigen fragments and finally it will be presented over the surface of the cell, forming the major histocompatibility complex, antigen complex. And this complex will be presented to the T lymphocyte. Okay, and next coming to the steps of CMI. So, firstly, there will be antigen processing and presentation, and later antigen recognition by the T lymphocyte, and then T lymphocyte activation and proliferation, and finally, there will be attack phase. So, in this, uh, okay. So, first of all, when an antigen affects the cell gets infected, and there is this MHC will gets binded with this antigen fragments, and later the complex which is formed will be presented to the T cell receptor, and finally this T cell gets activated. So, in this case, the MHC type is 1. This type 1, as we know, will present to the CD plus 8 cell. Okay, in the same way, here the antigen is again infecting the cell, and later the MHC is getting presented over the surface of the cell. But here the case is MHC2, and this MHC2 will present to the CD plus 4 cell. Okay, so after this, what happens is that the T cell gets activated and it starts proliferating. The CD plus 8 cell will form suppressor cells and cytotoxic cells. Okay, so these suppressor cells helps in regulating the cytotoxic T cell. Why does it regulate? Because when the activity of cytotoxic cells increases in the body, then the body loses its ability to differentiate between the normal cell and the foreign cell, and then the normal tissues might get damaged. So, to avoid this, the suppressor plays a very important role in regulating the activity of the cytotoxic cell. Coming to the function of the cytotoxic cell, it is also called as killer cell. It can kill the antigen in three ways. One is perforin mediated, and then by the release of the cytotoxic substances like cytokines, and then by the induction of apoptosis by the tumor necrosis factor. So this perforin will get embedded in the cell membrane and it makes the hole in the cell membrane, due to which the water enters from outside to inside, which is called as osmosis, and then the cell membrane gets expanded and finally it ruptures. Okay, and next coming to the CD plus 4 T lymphocytes. When these gets activated, again two types of cells will be formed, that is helper T cell and memory T cell. So this helper T cell is again of two types, that is helper T cell 1 and helper T cell 2. So these helper T cells produces interleukins, where HT cell 1, that is helper T cell 1, produces interleukins 2 and interferon gamma, which helps in the activation of cytotoxic T cell. Okay, so again, helper T cell 2 produces interleukins 4, 5, 6, and 10, which helps in the activation of B cells, which produces humoral immunity. So this is a place where T lymphocyte and B lymphocyte are cooperatively working together to fight against the antigen. So it's called as TB cooperation. And coming to the action of memory T cell, which helps in mounting of secondary response. For the first time, when the body gets exposure to the antigen, the primary response is very slow and is not much as good than secondary response. But when the same antigen affects the person for the second time, then the response is very good. That is, it provides greater response compared to the primary response. And this way there is attack phase. So this is how these processing and presentation of antigen occurs, and when the cells get proliferated, this is how they attack, by mounting, by release of interleukins, and by perforin mediated, cytokines mediated apoptosis. And this is how the cell mediated immunity works. So I'll be telling about the humoral mediated immunity in the next video. Thank you. Please subscribe.